Okay, whoa, that was loud. My mic is on, okay. Sorry about that. I think if you're not awake now, I hope you're awake by now. So welcome to this second day of our general meeting. Welcome to those of you who are joining us virtually. We're going to start with some quick introduction. I remind all of you that if you are experiencing any COVID symptoms, any ache, any fever, any headache, we would encourage you to stay home, to stay at your hotel. I see that there's more yellow and red tags happening, so just keep this in mind. And so I'm going to introduce this morning's speaker. Sorry, just making sure that everything is queued. And so first, for those of you that are joining us virtually, we encourage you to post your questions. Yesterday, we had over 700 participants. It's quite exciting. And thank you all for participating so actively online. And so this morning's session is all about trying to figure out how we can make the growing atlas that we're building more accessible to the general community, regardless of your domain expertise. We want to make sure that it's easy to query. We want to make sure that it's useful to the broader community. That requires a lot of thoughtfulness. And so to start having these discussions, we brought together many different thought leaders in the field. Our first speaker is Niels Gettingsburg, who's an associate professor at Harvard Medical School, and who is making his career and developing different visualization tools to empower researchers from a wide range of expertise to be able to explore data. And so, Niels, thank you.
Good morning, everyone, and thank you for the kind introduction, Chloe. It's great to be here, and I want to talk a little bit about Atlas user interfaces that can hopefully accelerate data access. And I submitted the title before I made the talk, of course, so things will change a little bit as we go along. But I want to start out by just giving you some background. Over the last couple of years, I've been working on a number of different data coordination center projects, starting with the 40-Nucleum Consortium, the DCIC, then more recently with the HubMap Hive. Last year, I became part of the NIH Cellular Senescence Network, or CENNET, and will be contributing to that data portal, and hopefully very soon we'll start work on the second phase of the Kidney Precision Medicine project, where we'll also be contributing. And so over the years, I've been thinking a lot about this question, right? So how can we design these user interfaces that make data access effective and efficient? And what do I mean by effective and efficient? So effective to me means it should deliver insight, and efficient means it should minimize effort for the user. It should remove friction. And if you want to take it further, we could also say, let's build data user interfaces that allow us to do science and do it quickly, because that's all what we certainly want to do. But to give you some examples of the kind of work that that I hope we can do in this community, I want to give you examples from HubMap and what we've been doing in the last couple of years to build the HubMap Atlas, if you want. And because nomenclature is always a bit of a challenge, as you know, I want to give you quickly the HubMap view of the Cell Atlas world. When I say Atlas, data portal, et cetera, just to clarify this for you, and in particular for those of you who are familiar with HubMap, there are two pieces really when we're thinking about HubMap. There is the HubMap data portal that contains generally the data that was funded by NIH, and then there is the Human Reference Atlas, which contains more the knowledge, so things extracted from the literature. In the data portal, you have functionality for data ingestion and curation. There are uniform processing pipelines. There are visualizations for molecular data, and a bunch of other things. On the Human Reference Atlas side, we have, as I said, knowledge such as the Common Coordinate Framework. We have spatial information about where samples were extracted from different sites in the human body. We have the ACT+B tables and many, many other things. And of course, we're working on bringing this together into a single unified resource. that would allow us to ask, important questions such as, what cell types are present where in the human body, how are they spatially organized, what information was used to establish that kind of knowledge, right? So that's where we need to connect back to the data. We want to know how reliable the information is, and we want to, really figure out how the knowledge was generated and derived from the data. So this is, what HubMap is overall in terms of data and knowledge products, but my focus today is really on the data portal side. And so what have we done to deliver insight and minimize effort? To deliver insight, we've created visualizations that we provide. We enable queries across data sets. It's a work in progress, but I'll show you what is possible right now. And we also integrate data analysis tools into HubMap. And so that is also work in progress, but I want to show you this nonetheless because it's really relevant to how we can accelerate data access and insight generation. When it comes to minimizing effort, I want to just briefly say and emphasize that we do this, in this case, primarily by building these user interfaces and the functionality directly into the browser or into the portal interface that is accessible through a web browser. So no need to download data, install software, anything like that. As long as you can open a web browser, in most cases your phone should be sufficient, although the screen is a little small for most things. You can do these things that I'm going to talk about. And so with the visualization tools, right, so our goal is to deliver insight, understanding of data, understanding quality, maybe generating hypotheses. So we have a number of different data types in HubMap. In fact, we have probably dozens of data types in HubMap, and they all require different kinds of visualizations. And you see a couple of samples here. So here, this is a visualization of a codex data set. You see this is actually the image that was originally generated that we're seeing here. You see wow, you don't really see all that much up there, do you? There is a UMAP or TSNE embedding. You see a heat map. You can see the controllers for the image, so channel controllers turning on certain markers, turning them off. We can also put a segmentation mask on top of that, map expression levels onto the segmentation mask and all that. So this is just one example for one data type. And if I can get to the next slide, which is always, here we go. We have other visualizations. So this one, unfortunately, the movie stopped playing, but I can assure you this is an interactive 3D visualization of 3D IMC data. Again, you have channel controls. You can rotate the data, and you can view it from different angles. You can slice through the volume, etc. Again, you can't see it. I really apologize. We have, in this case, here's a slide seek example where you're, okay, that also stopped playing, but you can, in this example, you see cell types that can be mapped to the spatial view, to the embedding view up there. There's a heat map. You can select marker genes and look at the expression levels of these markers on these visualizations. We can also, in top right corner, you can do standard single-cell RNA-seq with just an embedding view, some violin plots that show you expression levels, and then the heat map. You can see we can look at standard, H&E type stains. There's also an example where we have a light sheet microscopy in 3D as a volume that you can visualize in here. And all of this is enabled through the Vitesse visualization framework that we developed for this purpose, really with the goal of bringing the visualizations directly into the portal. So no need to download anything, no need to install any software. That was really our goal when we started this project. We wanted to minimize the number of clicks it takes you to actually start playing around with the data. And so all these visualizations that you see here, as I said before, are essentially one assay type, and they're one data set. So all of these are just a single data set. And that's only part of the puzzle, of course, right? So there's also the need to visualize multiple data sets. For example, all the single-cell RNA-seq data sets for kidney, or you might want to visualize multiple assays for a single sample, or both. So, right, multiple samples, multiple assay types. And that will require additional work. We have started that. We can load multiple data sets into Vitesse, for example, but when you deal with multiple data sets, multiple samples, you want to bring in metadata as well, for example, to stratify and compare information. Of course, and then at the highest level, we really want to visualize the whole atlas eventually. And this is the kind of work that Kadi Borner's group has already started on working sort of at the anatomical scale to visualize the atlas. We have some visualizations, for example, we can visualize our azimuth reference maps, which are also part of the atlas in Vitesse, but we really need to explore the space much more and really get an understanding, what does it mean to visualize a whole atlas? And so that's in terms of visualization what we can do today and what we need to do. In terms of queries across data sets, I just want to give you a quick overview of what's possible at the moment. And while this is live on our portal, I'm not going to tell you the URL right now because it is still working in progress. So this cells query interface allows you to select, for example, a type of marker, a gene or a protein, then you set the parameters for your query. So here I'm looking for cells that express UMOD at a certain expression level, and then we're looking for data sets where cells where there's a minimum number of like 10% of all cells in a data set have to express UMOD at that level, and then we want to find all data sets that have such cells in them, and then we get a list of results, and in these results we can look at the expression levels for UMOD in that given data set. We can also look at in which clusters or cell types UMOD is expressed, which cells actually match our query, which don't. And then, of course, we can open up the data set in the visualization. It's really just a click, right? So all you need to do is hit the link up there, you get the data set, you can then select the marker, highlight the expression of that marker in that data set. You can see this both in the spatial view and then also in this cell set or expression by cell set view at the bottom with the violin plot where this gene is expressed. In this case, it is highly expressed in a cell type called when we look at the cell types cortical thick ascending limb in this kidney data set, you can see the expression, and you see this yellow line down here in the heat map, that is expression of UMOD in these cells, and you can see when you compare this to the expression levels, that is the cell type. And we can then start exploring and interpreting the data. Is this timer 10 minutes or 15 minutes? Good question. Anyway, I'll keep going here. So in terms of data analysis tools, we want to help people go beyond what we can do in the portal. And what we focus on right now in terms of analysis in the portal is computational analysts who do cutting-edge work, right, who use unique workflows and tools to process data. We want to find a solution that is extensible and that can be contributed to by the community. We want to combine HubMap data with user data, and we want to use the HubMap infrastructure for computation and storage, and essentially allow people to pick up where the portal leaves off. And so we enable this by supporting Jupyter notebooks and workspaces in the data portal. And so, already today you can download a notebook, for example, from your dataset search that will link you to the existing data sets. You can do the same thing for any visualization. I'm being threatened here, so I will try to wrap this up, but I can't go faster than the animations. You can download a data set from, or the visualization for a data set as a Jupyter notebook, put that notebook into Google Colab, for example, execute the notebook, and you get the visualization in your notebook. You can start modifying it, add another view or something like that, and keep going here. And soon you will be able to run these notebooks directly in the portal through data workspaces that we're implementing right now, and then you'll be able to execute them as Jupyter Labs, where you can also upload your own data. So these things are very popular. We're not the first ones to do this. Crypto miners love these kind of things too, because it's great free resource, and so that's possible. I want to is there a way to fast forward through the slides? Probably not. So I will just say we can also support downloads through manifest generation based on the metadata that we have. That's possible in here. I'm just going to say that. And I want to wrap up with some takeaways. So one big takeaway is all of this UI work requires major coordination. So I'm just going to go a little bit faster here if I can. UIs and infrastructure really need to talk together, and there's a lot of talking that needs to happen between the people who build these things, and it goes beyond the UI and infrastructure, right? So infrastructure means data processing and storage, but also data analysis tools need to be aware of what we want to visualize at the end to generate outputs that actually can be visualized directly in the portal. So there is a lot of interaction necessary to make this happen. I was thinking a little bit about, how we're thinking about data analysis, interpretation, different tiers, viewing data, exploring data, analyzing data. I think what we typically do with data portals these days and cell atlases, we're somewhere between tier 1 and tier 2, right? Hopefully in the future we can build interfaces that allow people to build models and make predictions. That's where we want to go, but we're not there yet for sure. And finally, there are a number of challenges, right? It's Wild West times, sociological challenges, technological challenges, and then scientific challenges as well. I'm not going to go into the details here, given the timing, and I think what I do want to end up with, though, there are lots of opportunities for the community to get together, collaborate on shared challenges in Atlas user interface development, right? To share use case personas and other research artifacts. We should agree on common data schemas and formats. There are already some work in progress, funded by various funders and supported in the community, but we can do probably much more of that. I think we also should consider APIs as user interfaces, because many people here in the room are probably interfacing with data through APIs rather than UIs, and so this is really important that we do a good job with those as well. There's more. What else can we do? You can tell me now in the remaining 45 seconds, or you can apply for a job in my lab. That's definitely one thing you can do. Thank you. And
Neil, thank you so much for such outstanding talk. We don't really have time to take questions. I don't know, was there any questions online? But please find Neil's in the coffee break. One of the questions that I have for you that I hope we can have during the discussion of the day is metrics of utility, how you get feedback from users and so forth. So perhaps we can have this conversation during coffee breaks. Thank you so much for this outstanding talk. Should I go? Okay, so the next presentation is going to be given by a team, a tag team. Jonah Kuhl as well as Arathi Mani. Jonah is a science officer lead at CZI, where he leads the single cell program, while Arathi is a software engineer at CZI. And so we look forward to hearing your thoughts. You have slightly more time because you're a duo.
Slightly more time, but not too much time. Well, first let me start with just saying how nice it is to see everyone. This community over the last couple of years has made incredible progress, and it's been great over that time to meet new grantees, new partners, and to see all of you in person, to be with those of you that are able to join virtually is really a treat. So thank you to the organizers, thank you to all of you for being here. It's really exciting, and I know Arthi and I are really looking forward to sharing what we've been up to over the last couple of years at the Chan Zuckerberg Initiative. So let me start with a single slide overview of the CZI single cell biology program. And I think many will be familiar with various efforts and funding programs that are kind of summarized here in our little schematized single cell. Starting off with the seed networks that started about 3 years ago and then have evolved and most recently now include the dedicated efforts around ancestry and pediatric networks. And the goal, or one of the key objectives that I know we all share, is to support and as a community dedicate ourselves to the generation, assembly, and use of healthy reference single-cell atlases. And in this context, the Human Cell Atlas and this community has been critical for CZI and really been a lighthouse project for us since our inception about 5 or so years ago, when Priscilla Chan and her husband Mark Zuckerberg launched CZI Science, and really exemplifies many of the areas that we see as being most important to us, and in particular building fundamental references and fundamental tools to support this work that then can make it possible to cure, prevent, or manage all diseases, which is the mission statement that they set forward for CZI Science. Now, the other thing that people will be familiar with in these programs is that a large focus of them, including the seed networks, ancestry, pediatrics, to a lesser extent inflammation, have really been focused on the generation of data or the development of the tools necessary for that. And all of them, to kind of varying degrees, have support for and requirements for the inclusion of computational biology within it. But I think as we will appreciate from Sarah and Aviv's kickoff yesterday, Donna and Fabian's talk, the computational challenges are far too numerous and far too great for this to be even close to sufficient. And so what Arthi and I are hoping to touch on today is really how we're looking at funding, building, and partnering around the data ecosystem to support the continuing efforts and really the integration and the march towards delivering and increasing the utility of these reference data sets, not just from the CZI grantees, but the community at large. And so everything we'll talk about is very much open, it's available, it's intended to support that work. So what we're going to do is we're going to talk about efforts, again, from the last couple of years that we are funding. Arthi is going to talk about the work that we are actively building, and then also some partnerships and hopefully opportunities to work with folks like Niels and many others that I think you'll hear about through the session. Okay, so let me start with the funding part of it. So the funding of this data ecosystem, I think, falls into at least 3 different areas, and 2 of these are brand new, so I'm really happy to announce the funding for a couple of these efforts. So the first, of course, is the support for collecting all of the data that's being generated across the ecosystem. And this, from the very start, has been something that we and other funders really care deeply about. And it's now, after 5 years, time to kind of reconnect and look at the data coordination platform, which I don't think we've spoken about too much today, and happy to announce that we've just extended the DCP development support for an additional 3 years. And I think there's a couple folks from the DCP here. Maybe you can raise your hands and wave. Yep, there you go. Looks like that side of the room. Yeah, hand over. And so the progress in the data coordination platform has been great, and also a lot of lessons learned. And one of the key lessons is that the priority coming forward is really the storage of raw data, and in particular managed and controlled access, which hasn't been supported. And so I would strongly encourage all of you to connect with them, give them feedback, let them know what's going on, and to kind of create those inputs and discussions so that this can continue to be a really successful part of the project. The other new grant is around data integration, and this is an effort that will help support and connect with the bionetworks. And so as these roadmaps that are being developed, as the data from those groups are being prioritized, we'll provide dedicated support for a subset of those to really help accelerate and also to connect and hopefully create a framework that can be extensible to additional bionetworks. And help to deliver and get to those integrated reference drafts that the community is working towards. And then finally, this middle category of more of a distributed opportunity for funding that we will continue to do. So it's at least 3 cycles. We've just completed one and launched another one, which I'll talk about in just a minute, that we've called Data Insights. Credit to Sarah here as we kind of talked about this idea over time, and really focused on using currently accessible data to develop new tools, harden existing tools, and gain insights from it. So this could include, again, different meta-analyses or integration efforts, but also refining the tools and different opportunities throughout the community. Okay, so let me briefly talk about Data Insights. As I mentioned, the second cycle is now open. You can find information on the website. It's open for a couple months, so again, everyone is free to apply. Some example areas, just to give you a sense: meta-analysis or integration of different data sets that are maybe outside of bionetworks or maybe would represent a nice opportunity for new bionetworks to come together, as well as trying to really push forward in some of the different spatial methods that I think we got a sense for yesterday as being exceptionally exciting with a lot of promise and also a lot of needs for computational work. Okay, now let me very quickly touch on the partnership part of things. Again, as I mentioned, our hope is to support our grantees, but really support the community at large. And in particular, I think there's a couple things that are really exciting. So the first is I know others have been really invigorated by and excited by this work that has been put together by a great group in the Open Problems for Single-Cell Analysis. And I think now have completed a really successful competition last year on multimodal single-cell data integration that I believe Fabian mentioned yesterday, and really building bridges to the wider computational biology community so that they can come in, understand how awesome these problems and challenges are, and bring their expertise. And then the second is kind of broadly a call to reach out, get in touch, and identify ways to collaborate and build community around some of these problems. So this is a photo from a recent workshop that brought together members that have generated data in the bionetworks for immune and kidney, as well as various NIH efforts such as KPMP, HubMap, et cetera, as well as groups that have developed different integration methods to start to work on the technical challenges that are going to be important to integrate and help drive forward those projects. And then I will also just kind of add a quick note that a lot of excitement for collaborations more broadly. So I think you'll hear from Arthi about what we've been building, and I think there's great opportunities to continue to collaborate and connect with efforts such as the cell annotation platform that I think you'll hear about from Evan, as well as ASCTB and from Caddy, various portal interoperability in these data working groups, as well as data hosting. So kind of an open call that when we talk about Cell by Gene, it's really meant to be a community resource that anyone is welcome to contribute to use, et cetera. Okay, with that, I'm going to hand it over to my colleague, Arthi.
Thanks, Jonah. So Jonah just covered some of our partnership and funding efforts, and for the rest of this presentation, I'm going to be talking about the build efforts of the single-cell technology team. And my hope is that you all leave knowing that if you're in need of standardized single-cell matrix data, you'll hopefully check out Cell by Gene portal. So the suite of tools that the tech team works on building is called Cell by Gene. Our aim is to build tools to empower a broad variety of scientists with a broad background in computational skills to make use of single-cell data. It consists of two platforms. The first is a suite of hosted tools called Cell by Gene Portal. Cell by Gene Portal is built on single-cell data that have been submitted by scientists, including many of you all, that has been curated by our data curation team and adheres to a minimal schema. It also contains a suite of visualization tools to empower scientists to form hypotheses about others' data without actually having to download the data itself. The second platform is called Cell by Gene Desktop. Cell by Gene Desktop is like a pip installable desktop application that enables scientists to explore and annotate their own data sets. For the rest of today's talk, I'm actually going to be focusing on Cell by Gene Portal, as that's where the majority of our releases have been over the last couple years. Also, you'll have to excuse the really shoddy logos here. We're actually in the process of a rebranding effort, so this is what happens if you make an engineer create some logos. So this is an overview of the different components of Cell by Gene Portal. I'm going to dive into each of the blocks represented on this slide, except for the one on the left. Those are tools that we built specifically for our data curation team to enable them to more quickly submit data sets to our platform, as well as make sure that it's validated against our metadata schemas. So first, I'm going to talk about our data resource, which is the aggregation of all of the data sets that have been published to our platform, and walk through some of the tools to be able to search through it and then download data sets. So our goal with building the data resource is that every data set that gets added to it has been standardized with metadata has been annotated with metadata in a standardized way, and that it contains a minimum set of schema fields to enable downstream analysis as well as reuse and integration. Our fantastic curation team featured on the right side of this slide consists of folks from the Lattice team at Stanford, the HCA DCP, and Sanger. And these are the folks that work with labs and consortia to curate their data and make sure that the data sets are of high quality. And I believe that some of the folks from the curation team will be available today in one of the breakout sessions, 1.3, with the cell ontology workshop. So if you're interested in submitting data, please check them out there. And we're super proud of the work that we've done here in partnership with you all and our data curation team to be able to now offer the largest standardized collection of single cells. And to give you a sense of what this volume looks like, we have 32 million cells in our data resource, 24 million of which are primary cells. That 8 million cell difference are cells that have been reused as part of meta-analyses. For example, the azimuth reference, which is also published to Cell by Gene. They come from 413 data sets, which are part of 72 collections, and they represent 6 modalities, 186 unique tissue types, and 515 unique cell types. And then to give you a little sense of our current user volume, we see about 90 unique visitors who are downloading data sets, about 300 to 400 data sets each week, and we have about 1,500 visitors to any part of the Cell by Gene portal today. And if you're interested in visiting the portal yourself, you can go to cellbygene.cziscience.com. This is a screenshot of our collections page, and the filters on the left sidebar are enabled by the standardized metadata that we collect. So using that, you can filter down to data sets that you're specifically interested in, and you can download it in either the ANData or Seurat file format. And then I'm going to briefly talk about our two visualization tools. The first one is SC Expression. SC Expression is built on the 24 million primary cells that we have in our data resource, and it enables you to explore and compare gene expression across cell types and tissues. Similarly to the collections page, you can use the left sidebar to filter the gene expression down based on our standardized metadata, including by sex, ethnicity, and disease. You can also filter the expression down based on specific data sets that you might be interested in. And then the second tool is called Cell by Gene Explorer, which is intended to visualize single data sets that have been published to our platform. It enables you to dive deeper into the biology measured by single-cell experiments and explore states and signals that might differentiate cells within a type. So you can see here, Cell by Gene Explorer has been loaded with the Tabula sapiens dataset. And it's intended to not require any computational experience to use the tool. You can interrogate a data set using features such as coloring by cell annotations or gene expression. You can also perform differential expression analysis on it, on any two custom clusters of cells that you select. And you can color by the gene sets that are resulted from it, or any genes within the gene set. And then last, I'm excited to talk about one of our newest efforts called SOMA. So SOMA is a new project that's intended to address one of the growing needs in this ecosystem, and I think it's a common theme, in especially today's day of the conference. We see that data is increasing in both size and complexity, and we need tools to be able to easily manipulate large multi-data collections of tens of millions of cells, hopefully not too long from now, hundreds of millions of cells. And the data resource of 30 million cells is actually a good example where we can't actually offer it as a whole, at least in one of the file formats that exist today. So SOMA is an API standard that is flexible, open source, extensible, intended to access data in a variety of file formats, also enable data to be put into a cloud-friendly format. And then using the API, you can query, slice, or stream the data without actually having to download the whole thing itself. So this is an active project in partnership with TileDB. TileDB is helping implement out the first implementation of SOMA API, and we have a parallel project to offer the data resource backed by this implementation of SOMA. So if you're interested in keeping up with the progress of this project, please do sign up for the mailing list using the link on the right-hand side of this slide. I also have a little demo on my laptop, and I'm going to wrap things up. Yeah, perfect timing. I just want to send a big shout out and thank you to the entire single cell team, many of whom are here today. This work that Jonah and I presented today really wouldn't have been possible without this entire team. And I also want to say a big thank you to you all. Your input and feedback is absolutely critical in helping us build tools to help facilitate single-cell biology research. So please don't hesitate to reach out, and thank you.
Thank you both for a fantastic talk. Do we have questions online? Maybe some very quick answers for both of you. So I guess I was going to ask you how we can, interact with your team to think about new features. So I guess the best way would be to email and set up a meeting, or? Yeah,
the best way, cellbygene@chanzuckerberg.com.
Perfect. There's a question.
Yeah, really thanks a lot for all the community building you have facilitated. Certainly for our community, it has been instrumental to get the data up and quickly accessible via the Cell by Gene instance. Really great. One feature which we find sometimes helpful is to have a fixed time window where users can come and can interact with developers to ask real-life questions about interrogating the data on the platform. We call them office hours or something. Do you have any plans to offer such a kind of standing support service to the user community?
So I don't know that we have plans to do so. However, if you reach out to us, and I think Pablo is here. I don't know if you want to raise your hand, Pablo. This is something that we would absolutely do. Yeah. Currently it's been more kind of like one-on-one when people reach out. We're more than happy to do a seminar, spend some time. But if there's kind of broader interest, I think office hours or something like that would 100% we'd be.
Perfect. Let's talk.
Yeah.
And Jonah, thanks a lot. Great stuff that you do, really important for the community. Just super shortly, how about the spatial side of things? Everything based, Mapari, what do you think about the test, like the other things? Just some comments from your side.
Yeah, so the spatial side of things. So one thing to say is that among those 6 modalities, that includes spatial data. And I think a great example is the work that Ed Lean presented on yesterday within BICCN. So I know we're hosting data for Xiaowei, a number of different spatial transcriptomic, Visium sorts of technologies. So I think that's an area where clearly it's kind of the tip of the iceberg. And so we've already gotten a whole bunch of feature requests and trying to develop more there.
Within Cell by Gene?
Within Cell by Gene, correct. And then I would say, yeah, so that's kind of one, is reach out to us. I'm sure that it's kind of feature deficient, but would love to learn more. And then the second is back to that data insights question. There, we've kind of called out opportunities and trying to encourage more tools development, meta-analysis with spatial data sets. So I think those are kind of 2 opportunities where we could help support it, but then also appreciate that there's a lot of work to be done in that ecosystem and would like to fund and learn more with you all. Yeah.
Thanks.
Please join me to thank again our speakers. Thank you. So our next speaker is Nir Yosef. He's an associate professor of electrical engineering and computational science at UC Berkeley. He also joins the systems immunology department at the Weizmann Institute. Nir is known for all of the great computational tools he's developing to query large amount of signal cell data sets, including my favorite cells, the immune cells. So I look forward to hearing your talk, Nir. Thanks.
We have a remote here? Or? All right. Well, thank you for the introduction, Chloe. So I'm going to focus today about the Tabula Sapiens project that, as Aviv and Sarah mentioned yesterday, that the major release happened earlier this year. This is a joint effort for many groups in the Bay Area between Stanford, BioHub, UCSF, Donor Network West, and my group at UC Berkeley. So it was a large effort, right, with a draft of the Human Cell Atlas from 14 donors with close to 500,000 cells and 24 different tissues. And after a lot of curation effort and manual inspection of this data and some use of algorithms, also an annotation of about 400 different cell types in this data set. Right, so what do we do now that we have this data set out? So one thing is to have this portal that people can access, and it provides all sorts of views to the data with cell by gene, data downloads, and so on, and other tools. And that really provides a way to look into this data and start understanding it and start browsing it for people in the community. But now that we have this data set in our hand, right, and in my group we are sitting and thinking about what can we do with it, right? So we have this reference atlas in our hands with all this annotation. What are the challenges? What are the things that we can gain out of it? So I'm going to share with you 2 things that we started developing for making use of this atlas. A third one I would just mention very, very briefly if I have time. But the idea is to really leverage this information to do new stuff. So the first thing is almost a classical problem now that people have mentioned here multiple times, is the problem of annotating a new data set with the reference atlas at hand. Right, so this is a problem that we've been working on in my group and other groups for quite a while, and the idea is that you have your query data set right here. Oops. You have your query set right here, you have your reference data set, and you kind of just analyze them together to make predictions for the labels of your query of your unannotated data set. And there are many different approaches to this problem, but by and large, 2 of the most prevalent approaches is to try to integrate the 2 data sets together, put them on the same space, and then make predictions by who are the neighbors of an unannotated cells, which are the annotated neighbors of it. Another approach is to build classifiers, a more classical machine learning tools is just to use features to train some machine like a random forest or other approaches and use that to make predictions. And both of these approaches have been used by various tools. As you can see here, for example, for the integration, we have a CVI, ScanVI that Fabian mentioned yesterday, Scanorama that Bonnie Berger mentioned yesterday, and so on. And on the predictions, things like support vector machines or random forest. So if you look at the various papers that do comparisons between all these annotation methods, you can see that there is not really one size fits all. In some scenarios, one method is better than the other, and there are all these discussions and so on. So what we decide here to do is to take a more comprehensive approach and take a meta-analysis approach, if you will, to democratize the annotation process, right? So it's a new tool, we call it POP-V, as in popular vote, and the idea is to employ a set of algorithms. So we have 7 different algorithms that are currently all part of this one big pipeline, that all of the algorithms basically get a chance, given the tabula sapiens's reference, given a user query, to make predictions of what will be the annotations of each cell in the query. And then we get, instead of one prediction, we get many predictions, and then we are doing some consensus statistics and agreement statistics on these different predictions. And the idea is that generally, the annotation process, right, you can automate it only up to a certain extent, right? And the idea is to look at the agreement between the algorithms to see where we should really focus and where we should actually manually inspect the data. So just as an example for that, we took the lung part from the Human Cell Atlas, from the Tabula Sapiens project, and we took a new data set of a lung cell atlas from Mark Resnow's lab, and we wanted to see if we can actually re-annotate the lung cell atlas. So we've done that, right? So we were able to apply all these algorithms and get some consensus prediction. But in addition to that, we can also see what is the level of agreement between the different algorithms. So it's a score from 1 to 7 of how much the algorithms agree between themselves on every cell type that we have there. Because this data set has already been annotated, we can evaluate the results, and we can see that the consensus reaches quite satisfying levels of accuracy, but more importantly, as the consensus level increases, the accuracy increases. Now, why this is important? It's important because the places you want to look at, again, manually, the manual part is a very, very important part of the annotation. It's a very important part of the annotation protocol generally, and you can see here in red, these are the parts, these are the cells that are really marked, really highlighted as the cells where the algorithms disagree with each other. And these are the parts, these are the places where the human in the middle, in a sense, this is where we need to focus. These are the places we need to look at. So in this specific example, we can see that there is a population here of intermediate capillary cells that was just not captured. It was annotated initially by the hand curation from the lung cell atlas, but it was not captured by any of the algorithms just because it falls into this border area, if you will, between two distinct and different cell types that we have, the annotations that we have there, right? So this is exactly the example that we see of the annotation process, right? You start with the automation, you have all these tools that you apply, but then you take the next step and see, okay, where do I need to look closely? Where do I need to actually use my judgment and do some manual work? So POP-V is available online now through the Tabula Sapiens portal. We just this week we're going to put let's say the production version. We still have a trial version there, but it should be replaced in the next couple of days. It all happens in a Google Colab notebook. As a result from applying this pipeline, you can upload your data and get results. Again, overlaying it on the Tabula Sapiens project, all these views, the results of different algorithms, consensus analysis, and get the objects of predictions where you can then pull into cell by gene or to ScanPy or to SCVI tools for further analysis, inspection, visualization, and so on. So that was one. The second part is now, and Fabian asked this question, it was a good transition for me. In the Tabula Sapiens project, we really think about the next step here, and the next step is going spatial and getting some spatial mapping of tissues from human donors. And the question is now, how can we use, or whether can we use the single-cell RNA sequencing data that we have in the Tabula Sapiens project to enhance spatial data? So we can do it in all sorts of ways, and I'm going to mention one of them today, which is I think is fairly clear and comes from almost a limitation of the technology, right? So when we think about technologies that are based on RNA sequencing of barcoded chips, there is almost inevitably inside each spot, if you will, there are going to be more than one cell. In some cases, even dozens of cells, depending on the technology, but definitely more than one cell. Right, so that begs the question, this classic, the convolution question, right? So if I look at the given spot, right, what is the composition of this spot? How many of these cells are natural killer cells? How many of them are monocytes, and so on and so forth, right? So this is the deconvolution problem, the discrete deconvolution problem, and there are many solutions to this problem that people have been working on, and so on. But let me ask you another question. Let's say that I know that there are 35% monocytes in my spot. The question is, what is the state of the monocyte inside this spot, right? Because there are all sorts of, there's a whole gradient of monocytes, all sorts of types of monocytes that are there. So what are the specific monocytes that I have in my spot? So I can make smaller and smaller and smaller clusters, right, when I do the convolution, but at some point it's going to break. We are not going to be able to do the convolution with a lot of clusters. So that's why we developed this new algorithm. This is in collaboration with Ido Amit, which we call the SVI. So the SVI relies on all this probabilistic modeling with variational autoencoders and so on. And the idea is to really capture uncertainty in the data and reflect it out. So the SVI, just really briefly, it has two components in it. The first one is a model that acts on the single-cell RNA-seq data. This model is similar in some way to ScanVI, and what it does, it has basically, you can think about it as two components. The first component learns how to classify different cell types, very, very broad categories of cell types from each other, and the second one learns continuous axis of variation within every cell type, and gives us some probabilistic estimate of what is the cell state within every cell type, condition on what I'm seeing, and condition on the cell type itself. The second part of the model acts on the spatial data, and it gets a spot, and what it does two things. First of all, it gives us this pie chart, if you will, an estimation of the pie chart of which percentage of representation I have from every cell type in my spot. But then for each and every cell type, it also gives us what is the state of the cells that are coming from this cell type inside my spot, right? So it's a two-level analysis. It gives us a more refined resolution on the data. In a sense, what it gives us is that thing. For every spot, it tells me how many of the cells in my spot are of type C, for example, monocytes, and what are these cells expressing. So cell type specific gene expression within every spot as a way of this two-level deconvolution. And just as an example, this is unrelated to the human cell atlas, but just as a proof of concept, we took this mouse fibrosarcoma and analyzed it both with the 10x Visium and with 10x Chromium for single-cell RNA-seq and spatial analysis. And what we can see here is that if we do the deconvolution part, we can see that we have this outer layer of dendritic cells and T cells. But then if you look at the monocytes or macrophages, they're kind of hard to discern in this data set. And we can see that they are scattered throughout the tumor and the inner part of the tumor. So the question is, what is really going on there? But this is really where we can pull out the information on gene expression and start getting like a higher resolution analysis and view what is going on there. And with that, we're able to really isolate a monocyte-specific expression of this MREG population that was mentioned yesterday, right? These regulatory myeloid cells, and we can just localize these cells to very specific regions, these perinecrotic regions in the middle of the tumor that we can also identify with the H&E staining. And by doing differential expression, again, cell-type specific differential expression inside this data, we can also say that this niche that they make there is hypoxic, right? So that's an example for how we can increase the resolution of the data that we have there that is on a kind of an averaging scale, if you will, with these large spots, and make these cell type specific inferences of what is going on in the data. So finally, we have this. Am I out of time? Okay. So really briefly, the final thing that I want to mention here, we drill with one slide, is online access to advanced analysis capabilities that we also want to give out, right? So we had all these views that we've seen here for being able to access the atlas and look at the atlas and query the atlas and so on, but what about analyzing the user's data? This is a project that we have now going on, and it's really coming up. We call it SCVI MADS. And the idea is to make available to the user community or to the community the models themselves. And you can think about the model as a representation, as a compressed representation of the data. And our vision here is that with this compressed representation of the data, you can query your own data. You can make some inferences on your own data, for example, transfer learning with that, like Fabian mentioned yesterday, but do it with a very, very limited compute resources, internet bandwidth, or the strength of your processor, right? So this is an endeavor we're really going at quite strongly with the idea of really making these atlases and the use of them, the deep use of them, again, in querying your own data, more available to people, regardless of their resources. So, and finally, the atlas itself that we have, the Tabula Sapiens annotations, we worked very hard on them, but of course they are not perfect, and people here are going to look at that. They're going to probably have a lot of comments on those and so on. So the idea is, and what we really want to strive here is to drive a crowdsourcing component, if you will, of the annotation of the Tabula Sapiens. So in a sense of, instead of having multiple expert algorithms, we can have multiple expert annotators here. So we have some infrastructure for that, for people to upload their own annotations to us, and that's, again, through Cell by Gene, through the annotation tab in Cell by Gene. This is all available on the Tabula Sapiens website. It can be done online or offline, where the data exists on Figshare. But just as a transition here, I really want to point out here the cell annotation platform, which is the talk after me, or one talk after me, and we are really looking forward to interacting and to collaborating with the CAP as well to make this very important endeavor, I think, available in setting up a consensus terminology, ontology, and annotation for the Human Cell Atlas. So yeah, so with that, I will conclude. Just want to acknowledge the Tabula Sapiens steering group at Stanford BioHub and UCSF, and our collaborators on the spatial data analysis and on SCBI tools and funding resources, and thank you very much for listening.
Thank you, Nir, for a fantastic talk. I see lots of hands. I think we have time for 2 questions. Jose, please make sure you state your name and your affiliation, please.
Yeah. Jose Ordovaz Montañez from Boston Children's Hospital. Nir, I have like 20 questions for you. This is amazing. But just to actually build on that last part, this is what I was going to ask, if there was a way of actually better crowdsourcing the annotation and actually making it a more popular vote. What are some of the main challenges that you envision in actually the intake of different annotations, right? How flexible can these things be? Do they need to be within a cell ontology, or can you basically free text and then kind of mine what people are saying?
Yeah, so I believe Evan is going to talk about it quite a lot, but I believe that the main challenge is really the ontology, enforcing an ontology, because we made our life easy thinking about, okay, we have multiple experts that vote, but they all have the same ontology, the same vocabulary. This is where things start to get complicated, and I hope we'll hear more about it. That's why we are really so keen to collaborate with them. Yeah.
I know that everyone wants to ask questions to Nir, but I was asked to keep time. And so please find Nir in the coffee break. I saw 5 other hands. Nir, would you commit to staying next to the podium at the beginning of the coffee break so people can get to you? Okay, perfect. Thank you. Just making sure we stick on time. Thank you again, Nir. So I'm delighted to introduce our next speaker, Irene Papatheodorou. She's a lead at the gene expression team at ABI, where she leads the development of large-scale single-cell platforms to empower analysis, as well as associate the phenotypes with underlying genetics. Looking forward to your talk.
Yep. Thank you, Chloe. And it's really nice to be here and meet the community of the HCA in person. So today I want to talk about how to develop user-friendly portals for the Human Cell Atlas, and also focus on our work a little bit. So first, what are the different types of portals? Because there are quite a few different categories. So the way we think about it, at least, is that the first type is how to find and archive data, such as the HCA DCP portal, the data portal. And then you have the type of portals that are built around a project or a consortium to really enhance the collaboration and disseminate the output of that project. And for example, we have the Kidney Cell Atlas and others that are quite numerous actually, and they are supported by portals such as SelectGene and so on that were mentioned before. And finally, there are the portals that are actually knowledge bases that anyone can query. And I give just one example here, the Single Cell Expression Atlas that we developed. So knowing this kind of three general types of categories of portals, who are their users? So the way we think for the first category, find and archive data, is really, they're really aimed at bioinformaticians and data scientists who want to download the raw data. Then the collaboration portals that disseminate a project are generally aimed for domain experts and collaborators, bioinformaticians or bench scientists, cell biologists, and so on. Whereas knowledge bases that anyone can query are aimed for any research scientist or clinician who doesn't necessarily know how to interpret single cell experiments. So the kind of, there are lots of examples of archives beyond the DCP portals such as ENA, SRA, dbGaP Geo, and so on. Where anyone can find and download raw and processed data and perform their analysis. And the same goes for consortium portals. Users can interact with the data and collaborate, investigate within a project. For example, annotate cell type annotation platform that was mentioned before. It's a nice example. And then we have the knowledge bases where actually what we're trying to answer there is really queries to find the localization and abundance of gene expression. So basically such as the query that Dana presented yesterday, so where is my gene expressed and how does its expression change in the disease? And there are some portals being developed for this. These kinds of questions, so it's the Single Cell Expression Atlas or the Single Cell Portal from the Broad and Cambridge Cell Atlas as well that I will present later. So as I said, there are currently excellent solutions for archives and also for consortium portals. We heard quite a few here, including HubMap, Cell by Gene, and so on. But as the whole human integrated data sets emerge, such as the three that were mentioned yesterday, for example, like the Tabula Sapiens and so on, knowledge bases come more into focus with the potential really to increase the impact of HCA through them by making the data sets accessible to more types of users and of different fields and so on. So what are the ingredients of developing a knowledge base for the HCA? And obviously it's a lot of work because we need all these integrated data sets and the standardized analysis. It's not really easy to combine all these data sets and be able to ask the question, where is my gene expressed, in which cell types, in which part of the body, and also visualize it in an intuitive way. So also the other important issue is the annotation of the cell types. So we need to agree on ontologies, on terms, and so on, of the cell types, the tissues, the different developmental stages as well, as we will have cells in embryos, in pediatric atlases, and also the adult. And also the phenotypes, as we're moving more into how we want to use the reference data set into biomedical applications in the disease context. We need to be able to capture this and also use this metadata so that we can interrogate these rich resources. And finally, we need to have intuitive visualizations across the different data modalities that have been discussed here. So now I want to focus for the rest of the talk on what we are doing. So we developed the Cambridge Cell Atlas So this is a collaboration between my group at the EBI and also Sarah Teichmann's group and the cellular genetics group at the Sanger. So the Cambridge Cell Atlas is really powered by the Single Cell Expression Atlas, which is EMBL EBI's transcriptomics and resource for all species. So currently, the Cambridge Cell Atlas supports the same functionality as the Single Cell Expression Atlas. So you can do some gene and metadata searches. You can browse the data sets that match to the gene that you have queried. And currently we have just below 100 human studies overall, and this includes HCA, but also other human studies focused on diseases and tumors and so on. And we include 51 process studies from the DCP, although some of the HCA studies, I should mention, they come from controlled access archives such as EGA, dbGaP, and so on. And what we're working on now is annotating the integrated human studies with cell type ontologies and so on, so that we can integrate them in the Cambridge Cell Atlas as well. So I'm going to give you a little tour of a typical query, because I only have a few minutes. So let's say, as I said, we want to ask where a gene is expressed, and I picked a random gene here called NAPSA. So what we get is really a list of experiments where the gene is a marker gene. And here I'm focusing on the lung cell atlas. First we get the typical UMAP, where you can see on the left-hand side, I don't think I have a pointer, but on the left-hand side of the UMAP, you can see the different cell types, the cells colored by cell type. They're all ontologized. And on the right-hand side, you can see the gene expression, and that what you can read from that is really, it has high expression in, for example, type 2 pneumocytes. And then we're working also with some illustrators to develop these diagrams that we call anatomograms of the different body parts. So you have an option there to click on the anatomogram, and you can see the lung and the area where the sample was taken. You can zoom in, it's the alveoli, and you can zoom even further in and look at the different cell types, and then look at a heat map on the right-hand side, where that gene is obviously one of the marker genes of type 2 pneumocytes, but also, and then you can also explore the marker genes of the other cell types in that region, for example. So basically, this is how we want to present the data and reach out into clinicians as well as other researchers. And also I would like to highlight one piece of work where we are collaborating with pathologists and computer scientists from the University of Edinburgh under the Gut Cell Atlas project. And the idea here, our collaborators are working on a workbench, on a comparative pathology workbench, which is really an effort, a software tool to enable the pathology image interpretation. These are images from colons and Crohn's disease and healthy colons. And what we've done there is we integrated views of the single cell data from the gut cell atlas, where the pathologists can see which cell types the marker they are interested in is expressed right next to the different cells that they see on the image. So this is an application that we are exploring for clinicians. And in the future, we are discussing a future collaboration to add a spatial dimension of the HCA data in the Cambridge Cell Atlas, which is now strictly focused on single-cell RNA-seq. And we think that well, you heard yesterday we heard from Maz about the effort of the Human Embryo Atlas, so we will be collaborating with this group that uses a portal based on the Vitesse web framework to introduce these multimodal data sets into the Cambridge Cell Atlas. So I'm out of time. Okay. So, and that's my last slide, almost last slide before acknowledgements. So yeah, so how do we see the future? So basically currently the Cambridge Cell Atlas is powered, as I said, by the single cell expression atlas that is pan-species. What we're doing is we're basically looking for future funding that would enable us to develop further this human-specific functionality and the biomedical applications that are really out of scope for the pan-species resource. Finally, I would like to thank my team. So I highlighted with pictures 2 people from the team that are in the meeting, so Sylvie and Pablo, so please find them as well to discuss either ontologies, metadata, or analysis pipelines and web interfaces, and all our collaborators, the communities we work with, and the funders. Thank you.
That was a fantastic talk, Erin. That was great. Do you have questions online? We have time for one question. We'll take one question here to the right.
Hi, thanks for that. Anand Nandiyapan from Singapore. In terms of metadata to integrate into your existing data browsers, if you're going to move into phenotypes and other demographics, how easy is it to integrate that? Because the one level of visualization could be using different ethnicities or different phenotypes. Is that something which is in development? Or?
Yeah, so we do record phenotypes. We align them. Can you hear me? Okay. So what we do, we gather the metadata that accompany a study, whether they are phenotypes or also data analysis, metadata, or the protocol, and so on. And we annotate them with terms from the experimental factor ontology, as well as other different ontologies. And that really enables us to index all the data sets. And when we query, when we do metadata searches, the relevant data sets come up.
Thank you. And if you have more questions, please find Irene during the coffee break. Thank you. This was great. It's my, while he sets up, it's my pleasure to introduce Malte Luken, who's an incoming leader at the Helmholtz Center in Munich. He has been leading the human lung cell atlas data integration effort. He's also developing a lot of different single cell analytical frameworks and is a great contributor to the larger human cell atlas. And I think I'm going to keep talking while he sets up. We're running a little bit late in time, and so I'll be behind you making sure you stick to time. Just kidding. Half kidding. But I'm sorry we didn't have time to take as many questions today in this morning's sessions, but I do encourage conversation during the coffee break as well as throughout the breakout sessions this morning and this afternoon. Keep talking. Oh, we have a question online. No. Oh, please, I invite all of you that are joining us virtually to contribute to the discussion by submitting questions online. We have dedicated people looking at the questions online, and so we look forward to hearing all from you. And Dana Pierre has some thoughts to share.
No. I have a question to you, actually.
Oh, to me, okay.
Yeah, so, you are the, prototypical user of all these things that our panel is developing. So as a prototypical user, what would be your dream application?
migrate application, I think one of the biggest challenges is developing consensus annotation. I think all of us think very different about, I mean, to me, annotation is more than just tagging a cell label. It's actually how you go in to define a stable cell subsets. And it's not binary, it's not black and white. They're part of a spectrum, so we're trying to oversimplify it, which I think is going to fire back. And so finding a way to have a platform where the broader community that doesn't have computational background can actually effectively query to try to figure out if it is a stable cell subset according to their own definition and what type of label they would assign to it, so we can start having that conversation more broadly, I think is one of the main things I hope we can achieve. And we need to be able to bridge with their larger community that don't do single cell, and we need to try to reconciliate this with legacy knowledge so that we don't go and reinvent the wheel, and that's another level of a challenge. So we need
that
to think.
sounds like the flagship goal of the last talk.
Yes, I'm framing the last talk.
Oh, great promo for the last talk.
No, but I think beyond the last talk, I think we heard a bit about this throughout of the presentation this morning. And so I think having all these folks coming together, right, and brainstorming about how we can develop collectively some tools to empower the community would be great.
Since Malte is not, I think, ready yet, I'll comment on that. I think in immune cells, this becomes really prominent. First, because people can call the same thing by a lot of different names. They just say it's the cells of these class, and they express A, B, and C, like the X plus and Y plus and Z plus cells. And then you could replace X, Y, and Z with 3 other genes from the same response, and you end up with something that sounds different but is actually the same.
Absolutely.
And the other place where in immune cells this really comes to the fore, and was in Nir Yosef's example, is that it's actually a continuum.
Absolutely.
And these separations are not the right way of describing it.
Absolutely.
And we're going to see it with tissue cells that are not immune cells too, when they follow on a gradient. Priska talked about that yesterday in her talk. But if you go if you follow a physical gradient and you're taking from the 2 extremes, you think it's 2 radically different things. And as you fill in the gradient physically, you realize it's just a continuum. You see that in the brain too.
Yeah, and as we add other modalities, which you're good now, as we add other modalities, I think it will help refine this annotation, but figuring out how to include other modalities in these visualization portals will be important. All yours.
Thank you. It's great to have such experienced people who know how to fill the time with really interesting conversations while I set up. That's going to be a lesson to me for using Keynote on an old laptop, but hopefully it goes smoothly from here. It's a really great honor for me to be invited to talk today. One of the reasons I'm in the single cell field is because I saw a talk about 5 years ago in front of this background, and that really motivated me to join this great community, and it's been a pleasure to meet you all in person. We're talking today about reusing data, and one of the the purposes of a reference atlas is really how to repurpose published data sets to provide a value for the community beyond what the data sets were initially generated for, to try to get it a little bit more out. And we've heard yesterday in Fabian's talk and a couple other talks mentioned as well that we've recently built an integrated reference atlas for the human lung, combining 14 to 46 different data sets, depending on whether you look at the core or the extended, harmonizing reference, these cell annotations, metadata, and anatomical location. And then Dana pointed out something amazing, which I thought I'd put as a quote in here. An atlas is only as good as we can query it, right? So the question really is, why do we build a reference, and what are those use cases? And Anir spoke about this a little bit before as well, and there are very many things. We have very different expectations of an atlas, a lot of which have been mentioned already. Maybe some things that we specifically have focused on is building consensus. If I call a cell type a goblet cell, you might call a goblet cell something quite differently. And so only really by combining these data sets and then querying them specifically can we really build this consensus. In the generation or the building of the Human Lung Cell Atlas, we specifically focused on a couple of these to try to optimize the building process for the atlas for how we will later be able to query it. And these are just 4 of the examples of specific technical decisions we made to optimize for these use cases, including rare cell identification, providing context for disease analysis, and rapidly analyzing new data, as well as describing population variation. Given that I have limited time, I'll try to get to a couple of these, but I'm happy to talk about any of these later on as well. Specifically for optimal recovery of cell identities, and especially rare cells, There are many data integration methods out there. There are, when I counted last year, over 50, and I'm sure another 10 have been added recently. And these are all interesting methods which have different approaches, some more similar than others, but they will output different embeddings when you use them for integration. And we have found that there is a kind of a balance between the removal of batch effects and the conservation of biological variation, which these different methods can strike differently. So, using kind of our work from last year on publishing data integration, or earlier this year, we applied a similar pipeline that we built to try to evaluate what the different integration methods are doing for our data, for our lung case. So we took a subset of a couple different data sets from the human lung. Pre-processed them in different ways, because it's also not entirely clear what how the pre-processing affects this integration, and then applied a whole host of these data integration methods. If we had applied every single version of every single method, we would have gotten 68 different results, and we sub-selected a few here to be a little bit faster. And then we just kind of applied a battery of tests. So we developed a couple metrics recently to evaluate different aspects of batch integration at different resolutions, and a whole host of trying to encode intuition for what biological variation we expect to find. That will not be perfect, but that's the best we could do at the time. And so based on this, we find that ScanVI performed very well for our lung data. And now you might think, you know, a lot of these different results will still be biologically somewhat similar if you're, you know, using different integration patterns. And so we took a look into some of the more popular approaches. And indeed, with our ScanVI embedding, we could find very nice separation of rare cell types in the lung. And these all make up less than 1% of total cells and some 0.01%. And we find them quite clearly, nicely, kind of the ionocytes, neuroendocrine cells, and tuft cells represented differently. These are all supported by multiple studies in our core reference, and the original labels were not necessarily. And here I'm only showing kind of these cells which we finally annotated. There were a lot of cells which we annotations which we later regarded kind of as false positives as well. If we would compare this in just the tuft cell case to other popular methods, we found that kind of harmony embedding tended to split our tuft cells across different clusters, which enabled us was quite difficult to enable the recall there. And Surratt did recover all of them in one cluster, however, also clustered a lot of other cell types with the same cluster. We also found, now you may say, well, ScanVI uses some labels to optimize this embedding, and that is true. We used like a more semi-supervised approach here, but actually only used the rare cell type label and still found all these three subsets. We also found unannotated cells so something which the labels could not have told us about across this embedding, which worked quite well. Now, this is the case for the human lung. Because different methods have different balances between batch integration and biovariation, you could imagine that actually a different approach might be more suitable for a particular other atlas. And so you can reuse this pipeline again for other approaches, but of course, as new methods get developed, at some point the pipeline is going to be obsolete. Which is why, as Jonah mentioned before, together with collaborators at Yale, Chan Zuckerberg BioHub, Celerity, Imuni, we started this kind of open problems in single-cell analysis and have gotten a lot of buy-in from the community already as a living benchmarking framework. And we have started adding all of the kind of benchmarking data integration benchmark to this framework so that when a new method gets developed, it can immediately be added to the GitHub and be benchmarked with everything else, and you can use that pipeline to benchmark with new approaches on your own data sets, and thereby kind of trying to create a quantitative framework for how we can generate further integrated atlases, also including new methods. So our kind of solution for rare cell type identification has been kind of to benchmark data integration methods. But what about some of the other use cases? Specifically, we've also heard yesterday that SC Arches is a method we've developed within the TICE lab, which I've contributed to as well, to map new data sets on top of existing reference atlases. So if I have the human lung cell atlas, I map on top a query data set of something that, for example, Nir just showed as well, with kind of the Travaglini et al. Data set mapped on top of the tabula sapiens lung. Run it through an SC Arches model, which slightly adapts the trained reference model to kind of map this new batch on top, and thereby kind of adapting the batch correction. We can generate embedding of both of these data sets. Now, what I want to kind of get at a little bit more than what was introduced already is that there are particular criteria for how you build these models that improves their usability for new data sets. One of the things we found when benchmarking SC Arches is that the best performance for SC Arches models is when there are multiple diverse batches in the reference data. which means that if we have a single data set, which might only have sample batch effects, this will not necessarily train the model for understanding diversity between different types of batches when mapping new data on top. So really, it's crucial to have multiple different data sets, not all ideally from the same lab or from the same kind of setup, when generating an integrated reference atlas, and that generalizes it better to map also new data sets on top. Similarly, something which we found very useful with the ScamBI model is that it is end-to-end from count data. So using count data as input, it only requires minimal pre-processing for new data to be mapped on top. And this is something that actually I think was recently announced also for Azimuth, this ability to kind of allow new data sets with minimal pre-processing mapping on top of a reference atlas, and I think is very crucial for optimal usability. Now we tried this, of course, with a really, really cool atlas generated by the Teichmann and Meyer labs, which has kind of very detailed annotation of spatial multi-omics data sets with like way more annotations than we have in the integrated atlas, because we haven't been able to find consensus for all of these really detailed annotations. And when we have this in the same embedding, we can obviously transfer labels, as Nir mentioned, as was mentioned yesterday by Fabian as well. Now you may wonder, why am I just showing you how KNN classification works? This is a fairly basic approach. You know, you map a new data set cell in the middle, and thereby kind of can assign a label based on cell type A, even if it's slightly on the outside. But what's really interesting here is that if we are not certain, so the uncertainty in the label projection is a type of outlier detection which we can use actually to interpret our data beyond just the labels that are projected. And there are many different reasons why label projection might be uncertain. We might be doing a poor job of reference mapping, right? In this case, all cells from the new batch will have this problem, might be an outlier sample. But what's really interesting is that we might be highlighting directly cells or cell types which are not in the reference atlas. And if this reference atlas is a healthy atlas, these outliers are informative for further analysis to motivate as a hypothesis generation. And so when we apply this to the data set I just mentioned, we are pretty good a lot of the time. In some cases, this is like we're not sure what label to assign. And if you break this up, then we can show that we are most uncertain for data sets which are cell types which are actually just not in the reference atlas, which is exactly what we hoped to find with the uncertainty. But more interesting is that when we map cancer data sets from 4 different lung cancers to this atlas, a lot of the cells map quite well. Some don't map as well as you might, or as initially as you might want. But looking at the uncertainty of this label transfer, we find again the cancer cells obviously have a much higher label uncertainty, meaning that just by looking at the uncertainty without any further analysis, it highlights these cell types for further investigation because they're outliers from the healthy reference. We find similar examples in fibrosis, where we highlight kind of alveolar fibroblasts which are affected by a disease state. And several other examples in COVID, for example. But of course, the labels that we transfer are also relevant. So in this study, which we mapped data on top, there was a label of a tumor-associated endothelial cell. Now, it's not always easy to get matching healthy reference data for your study, for your disease study. And so when you don't have this, maybe the compositional differences will lead you to interpret a cell type, which is actually a healthy cell type, as a tumor-associated cell type. And we could label this actually as a venous endothelial cell because they map to every other cell type that we have across the board from other studies, but not necessarily from that study. And this has helped to kind of contextualize both healthy and disease data sets using reference mapping. So in summary, I'd like to review that we use kind of benchmarking data integration to optimize cell type recovery with SC arches and label transfer uncertainty. Have really tried to provide use cases for this reference. As an outlook, open problems is more than just benchmarking data integration. We are trying to quantify open problems in single cell analysis across different tasks, and we're very happy for as many contributions as people would like to get involved in. And I think the focus that we need to look at now is how to shift from cellular representations, now that we have these integrated atlas with lots of data, to representations of patients, because we are starting to generate the numbers where we can start to look at really this level of heterogeneity, And in the end, diseases manifest in individuals and not in cells. So that will better enable us to understand this diversity and disease manifestations as well. I have to thank a lot of people. Lisa, who drove the Human Lung Cell Atlas amazingly, Fabian, who enabled a lot of this work, and Sasha Martin, who were kind of the biological backbone. It made sense we are not driving too far off, as well as a lot of other people who contributed data and knowledge for annotation specifically. And if you may allow me very quickly, I'm starting a lab at Helmholtz Munich, and we are hiring at the moment. So if you're interested in then kind of computational approaches for building and using reference atlases towards clinical applications, please get in touch with me, send me an email, drop me a Facebook message. Yeah, thank you.
This was a great talk. Thank you, Malte. We have time for, oh, there's a lot of hands, but we only have time for one question. I guess to the right. Please state your name and affiliation.
Shyam Prabhakar from the Genome Institute of Singapore. Very nice overview, Malte. I was curious about one thing you showed, adding new data to an existing atlas, which is a very important problem. And I think the green dots you showed in that UMAP, they seem to be confined or consigned to the margins of your UMAP. And we see that too quite often when we integrate data. How does one ever know whether that's really a batch effect, or is it really that new data set is occupying just a very small fraction of the expression space of your reference?
Yeah, that's a great question. So just to be clear, the image that I showed with the green dots, it's because they're plotted on top for easier visualization. There is actually quite good overlap with the reference atlas. Now, of course, you will have cases where it doesn't map quite nicely, right? At the moment, one of the things we're using to assess kind of what that might be is this label transfer uncertainty. Now, based on KNN classifiers, there's definitely ways to improve, and we have worked on different approaches to also, not just use KNN approaches, which might be more reliable. But if an entire sample, all the cells from a sample have high uncertainty, we might think that has something to do with the residual batch effect. If it's a whole data set, definitely residual batch effect. But if they are individual states or different cells, while others map very well, we start kind of getting a little bit more attuned to like what is really happening there. And that's something that merits some further further queries and kind of looking into it a little bit more detail with clustering, marker gene detection stuff, anything that you would typically do for interpreting your biological data.
Thanks, Malte.
I know I said only one question, but we'll make an exception for
I've got an exception.
Yeah, go ahead.
I have no idea why. Malte, I have a question that's not related to the main part, but when you talked about the benchmarking of different tools, how carefully have you thought about the benchmarking data? So the one thing that bothers me about a lot of benchmarking studies, and I love them and I do them too, so it's not a criticism, is that in fact when you compare methods, it really depends on which data you're comparing them. And we often tend to aggregate around particular data sets, and we benchmark so closely on them that we're basically overfitting to the particular, not in the traditional sense of overfitting, but to the particular biological nature of the system that they represent. You see this a lot with trajectory methods. You go and you read a paper that compares, and people do a ton of work for it. But in the end, if the system underlying the thing they're looking at has particular nature, yes, some methods will perform better for these purposes than others.
That's a super good question. I feel I have a really cool answer to this as well, because there's two parts, right? For the benchmarking data integration, we try to just take as complex, different aspects of complexity as we could, right? That's kind of the thing everyone tries to do initially, right? It's just like, how do we get as representative problems as we can for benchmark data integration. That's why we have 13 different tasks which we benchmark data integration methods on. This will not cover everything. So for open problems, what we're doing is this is an entirely open community, right? So it's essentially centered around a GitHub where anyone can add a new data set, anyone can add a new method, anyone can add a new metric, and the idea is that there will be discussions about exactly this, because somebody will say, oh, I think this data set is really important, my method performs really well in there, you add it, but then there's also 5 different data sets. And so as soon as you aggregate a lot of these different data sets, then there can be discussion and meta-analyses about the benchmarking. So who benchmarks the benchmarks? And I think that's something which we are uniquely set up to do in open problems, once you aggregate huge amounts of different data sets for benchmarking and answer this question.
Thank you. Please join me to thank Malte. I'm sorry for all of you that didn't get to ask your question. Find him after. Thank you. So while we transition presentations, it's my pleasure to introduce our last speaker of this session, who promised me to stay on time, Evan Peterson, who's a computational biologist at Harvard Medical School and the Broad Institute, who's leading the development of the cell annotation platform, which is a centralized platform that will hopefully help empower our community to kind of work towards a consensus cell label. So,
precisely.
Evan, all yours.
Chloe has been very intimidating with the other speakers, about 10 minutes. So 10 minutes, I can do this. Okay, so thank you very much. I'm honored to be here, and thank you for the self-promotional comments about CAP from Nir, Dana, and others. So I want to give an update about where things stand with the cell annotation platform, basically review the problem that we've been discussing here in this session with annotations, give some details about our current release, and then discuss some upcoming features, and basically ask you, the community, to help us prioritize these features. So I think everyone here knows that annotations are a problem, but I just want to make sure that we're on the same page about the nature of this problem, and it's a fundamental problem with the annotations themselves, I would say. Let's say that, let's do a thought experiment here, and I were to give each one of you as homework a process data set. It had the same clustering, and I would ask you to go home and try to annotate these data sets. What's the possibility or the probability that each one of us would come back with something that's even close to a consensus annotation? I'd say pretty much slim to none, right? And the issue is really fundamentally we don't agree, we don't have the same vocabulary for these annotations. We call them different things, we have different definitions for these cell types. But also, we would come back and there'd be different resolutions for these annotations, right? So we need some sort of system to accumulate these annotations and somehow come up with a way to reconcile the differences. The theme of this session is actually that we'll have the individual biological networks try to do this at a large scale. So this is a problem, and many PIs have been thinking about this problem for some time. Right, it's mostly been spearheaded by Peter Karchenko, and there's others here as well. And so there's also been quite a large team that have been working on this, and I'm very proud to show them now to you all and to put their contributions at the front. Software engineers at Harvard Medical School, David Asumi Sutherland at MBI, leading the ontology efforts, and of course some crackerjack postdocs in Fabian's lab. So the solution that we've been working on is something called the Cell Annotation Platform. It's really a community-driven marketplace, I think is what we'd call this, for users to create, explore, and store these annotations. It's basically some sort of way that we can create a standard for the community for creating these annotations, uploading them, editing them in some sort of standardized way, publishing them for others to use, and then allowing the community to have some sort of medium to disagree with these annotations, to upload their own annotations, to re-annotate other people's annotations, and somehow come to a consensus for these annotations. So I said we have standardized APIs formats, DUIs upon the publications. We try to version these, hopefully as we aggregate data, we'll be able to come up with some sort of reference for these cell identities. So the four main components I would say for CAP at the moment is it's a data repository at heart. It's a database. There's a functionality to upload the annotations, edit and publish them. And the future steps will be talking about the annotation UI that we're building now, and after we aggregate enough data, the cell card summaries. So the site is online now. I checked this morning, there's about 10 million cells with annotations. You can take a look at the UI, the URL there. Just to walk through the current release that we have. There's sort of a sign-in user profile, ability to upload annotations, collaboratively edit these annotations in a private space, and then with collaborators that you can invite. Publish these annotations with a DUI, download the results, and browse and search other people's annotations. So just to walk through these very quickly, I think we're all familiar with these with the various social media platforms you use. You have a user profile that you sign into, and you can add your own metadata, where you're from, what's your institution, and so forth. There's also, it's very easy to actually upload these annotations. It's within seconds. It's basically a drag and drop modal, and we have a CLI that's up soon. Just to review, Peter covered this last year. The way that we're trying to organize this is very similar to GitHub. The idea would be that you have some sort of projects which are similar to folders, and you'd put annotations, multiple annotations with, well, you'd put data sets within this folder, right? This is what we're calling a project, and there could be multiple annotations within a single data set. When you do a publication, this is called a publication, there should be a version associated with this. The workspace is this something I'll cover very shortly. It's this collaborative user forum repo that users could use to standardize these data sets and these cell labels. And then for the cell labels themselves, there's quite a lot of cell-related metadata that we ask users to provide. So the publications, this is supposed to basically be analogous to a scientific journal article. It's going to be a collection of data sets with multiple annotations within those data sets. It's time-stamped. It shows who published this, collaborators, DUIs for those citations. There can be multiple versions. And as soon as you make a publication, it's available for the entire community, and you'll be able to download those files and those annotations. The workspace really is the sort of workhorse of the cell annotation platform at the moment. People would like a private locations. You can invite your collaborators to view and work with these annotations. As I said, it's very similar to GitHub or sharing, let's say, a Google document. You'd work together within these annotations using an advanced user form that sort of pushes you to give as much standardized metadata as possible, both for the cell types and for the data sets themselves. So you're able to basically specify which metadata in the data set is really the annotations versus some other sort of possibly irrelevant data set, metadata. We've hooked this into the ontology APIs such that there are nudges to encourage users to use some sort of standardized nomenclature, though it could be free text as well. In the case here, you can see someone types in IgA for the immunoglobulin A antibody, and there can be some recommendations that allows users to actually choose a term which is standardized, right? As I said, it's a private space for collaborations that you could use possibly coming up with the biological networks. For the cells metadata, basically for every annotation that you give, as in every sort of label for a cell type that you give, you're able to list the synonyms that are associated with the cell type, and then list what we think are some sort of category for these cell types as well. So, if it's a very specialized T cell, you could say this is a category of a T lymphocyte. So it could be as possibly as precise or as broad as you'd like. We also, if there are multiple annotations within a data set, you're able to view how those annotations relate to one another. So this is what people are usually calling hierarchical annotations. Usually if you call something with a very broad annotation term like B cell, there's also very precise terms like a naive B cell, a B memory cell. And this gets to a question that someone else asked. This is, why are we doing this? Well, if we ask users to give us this information, which is often free text or standardized terms from ontologies, we could start basically doing data mining with these terms, right? We could use NLP analytics and basic sort of text mining to see what is the consensus the community is using for these annotations. So that's the current release. You can look at the website now. For future steps, basically, what the current workflow is that you as the user, you have some sort of file format. At the moment, it's ANData and Syrah. We're working with Loom as well in the future. And you put in cell annotations within this file format, and you upload this to CAP. So the future that we're working on now is that let's assume that you have the molecular data, and it's not been annotated, right? So what we'd like to do is use the molecular data to allow some sort of real-time annotation predictions, something very similar to what Nir was talking about. So there's been other work in this space. SC Arches has been talked about, cell typist before me. And this is basically, we're including multiple sort of algorithms within CAP to do this step of cell type prediction. So you can imagine we are using all of the molecular data and all the text-based sort of data that we've aggregated to give this to users. And we put this in a UI such that users can see what are the outputs of multiple algorithms that are in this space, cell typist and so forth. And we also associate this with predictions that are based on the features, the distinct molecular features in the data set themselves, along with sort of majority-based terms that we see. This is an often occurring synonym for this cell type that you've included. And as I'm interested in talking to Nir now after this session, we'll be trying to include more models. So this is something, if you're working in this space, just let us know. We can easily add this to the UI. So in the next steps, by autumn 2022, I think we'll have the annotation UI working with the real-time cell type annotations. And then as we've started to aggregate enough data, we should be able to start including some more UI and data analysis sort of features. Niels' first talk was on v-tests. This is something we'll be working on. And if we've aggregated enough data, in principle, we should be able to start making these summaries, something like a RefSeq or a gene cards. These sort of what we're calling cell cards. So, I mean, my main raison d'etre for being at this conference is really to ask for more user feedback, right? I've had very little of this so far, as we've just released. So I think many people have ideas in this space. For instance, I'd like to see the evidence that you have for these annotations, or I'd like to see, maybe community ratings, or how can I contrast these annotations from user A versus user B. So I have ideas of how to implement this. Some of these ideas conflict with one another. So I really want to speak to you to see, how can we best do this. And there'll be a demo in, I think it's the next session after the coffee break. I'll give on CAP. We'll be sending out user feedback forms. And as I said, this really is an HCA driven project. This is your project. So if you do have any feature requests, please reach out. We'll be quickly putting those in now that we have the basic MVP structure released. So yeah, I'd like to thank the team that I have here. They've been spending quite a bit of time and effort on this project. Yanis Ilguzin, Maxim Svetlakov, Nick, Tigran, Kostya, and many others, and of course David here as well. And I think I'd like to thank Chloe, of course, and I think I'm within 10 minutes, which is good. And I think Peter Karchenko, I'd like to thank him personally, and he deserves a lot of credit for pushing this project. So, thank you.
Thank you, Evan. This is great to see that it's online. You launched it just for the general meeting. We have time for one question. Do we have a question online? Yes, we have a question here. Please state your name and affiliation.
Özgün Gökçe ile Munich, Oz. Thank you very much for all your work. And one question is how much consensus we need, like where to stop, because we have different modalities, different diseases, and we look to from different disease models compared. We will reach sometimes different annotations, and would it be especially continuous states? Could we actually compare in this atlas different modalities, different annotations that other studies used within the annotation network?
Yeah, it sounds like you had two questions there. One, are we able to actually compare the level of these annotations? And that's something that I think that we can basically do now. In terms of the larger question of how do you reach a consensus, I'm betting on if we get more and more expert annotated data sets, more and more data set, you'd be able to actually see sort of patterns emerging just based on the algorithms themselves, right? But I don't think there'll probably be a magic bullet to show that, at the end we have it's going to be a majority sort of process, I think. This is the majority label based on all of the algorithms. There should be a unique molecular feature. This is not a state, and based upon that, we'll be able to make these high quality atlases.
Thank you. We have one question from our virtual participant.
Yes, hello. This is from Maximilian Hausler from UCSC, who says, thanks, this looks useful. This website doesn't seem to have the expression data of the data site, though. To check an annotation, how can one find the expression matrix for a given data set on the website?
So the expression data is there, right? We don't actually allow users to upload annotations without the expression data. This is a very controversial decision we made very early on within this project. So you have to give us the molecular data, otherwise this whole process won't work. You have to actually publish this and be open as a community to sort of share this with others. So it is there. The real question is why didn't we show the UI for this, right, of the molecular data? And partially is that this is in the works, right? We just didn't want to give another UMAP embedding. I sort of felt that was a bit, yeah. So we're doing this. We do have the data, it's there, and we'll make UIs shortly.
But if others have feedback, Evan is here to get your feedback. We're also going to try to organize more demos across all the different bio-networks virtually in the months to come, so that you can all give your feedback and perhaps some more tailored solutions can be created for every bio-network. So thank you, Evan. Please also join me to thank all the speakers from this morning. Thank you.
Well, good morning, everyone. My name is John Randall. I'm the Chief Alliance Officer in the HCA Executive Office. I have a few housekeeping notes before we break for coffee, but I'm going to try to match Chloe's efficiency and get you into the coffee room as quickly as possible. The key point here is that we will be going directly from the coffee break into the first breakout session, and so you need to know, of course, where to go. We will post the breakout sessions in here, but they'll also be posted on screens out in the coffee hall as well. We are using 5 breakout rooms for this meeting, including the main hall here. One of the breakouts on computational querying will actually be in this room. We're also using the adjacent 2 rooms, so that's Heron's Hall to your left and Pralat's Hall to your right. And so good practices for tissue-based atlases will be over here, and then modeling genetic variation will be in Pralat's Hall. And then finally, there are 2 other rooms. One of them is the Hartman room, which is adjacent to the registration desk on the ground floor. And then the fifth room is actually in adjacent hotel space. And so if you are interested in going to the breakout group in the hotel space, we're going to ask that when the bell rings for the breakouts, that you actually meet the staff here. My colleagues Christine and Sabrina will be right here on stage, and they will then direct you over to the breakout room in the hotel. For those that are participating virtually, you are also very welcome to join the breakouts. We actually encourage you to join the breakout groups. You can do so by going back to the agenda and just clicking on the button for the right breakout room, and that will take you directly into the virtual space. 2 of the breakout rooms, be aware, will actually be a traditional Zoom meeting, and so you will see that marked by a Zoom in parentheses on the agenda. Finally, then, I think we'll begin the breakouts at 11:10. Here we go, they're up on the screen. We'll begin the breakouts at 11:10, so we'll have coffee until then. You'll hear the bells that we've been doing all along to get you into the breakout room. Please be efficient so we can get the breakouts started on time. At 12:35, we will end the breakouts. You will go directly to lunch. A couple of notes about lunch. So we have reconfigured the buffet, so there are actually 2 lines. So please be aware that there are 2 lines to try to hasten that process along. It's a beautiful day outside. It's cooler than it was yesterday, so we definitely encourage you to take your lunch outside into the tent and eat it there. I do want to remind people that no food and drink is allowed in this main hall space or in these adjacent 2 rooms. So please do not bring food and drink into those rooms. And then my last note is that from 1 to 2 p.m., the Helmsley Charitable Trust is hosting a networking hour, if you can call it that, a networking lunch for anyone interested in gut research. So this is not anybody who is a Gut Cell Atlas grantee or anything, just any gut researchers who are interested in meeting each other, networking, perhaps collaborating. That will be on the terrace between 1 and 2 o'clock. So those are my notes. And then finally, last but not least, please be back in this room at 2 p.m. for our keynote address from Elaine Fuchs. Thank you very much.